Okay. And you can see the slide's not that, there's nothing blocking it, is there?
Nope.
Okay, good. Great. Okay. So it's nice to be back here again this year. Miguel and I are excited to talk to you about the bicistronic program. We are going to kind of give you a little bit of a survey. I mean, we have at least one new late onset patient. So we're starting a little bit from the beginning. I'm going to summarize the program all the way through a little bit and then give you an update at the end. And since everything begins with the vector and the mice, Miguel's going to start, and I'm just going to run the slides. So Miguel, just tell me when you're ready and I'll advance.
Yeah, great.
Everybody, I'm Heather. This is Miguel, if you don't know us, by the way.
Great, thank you, Heather. So I'm just a warming act for Heather. So Heather, if you could get going. So this is a new generation of vectors that we've designed that encodes both genes in the same vector, which is something that we had been working on for many years, and so it can express all forms of hexosaminidase that are naturally occurring in the body. So hex A, that is a heterodimer to different proteins, hex B, that is a homodimer with the same protein of hex B, and hex S that has two hex A subunits. So this is the drawing of the vector here, which is a little unusual, but it actually works quite well. And if you could get to the next slide. So as Heather said, we always start experiments by doing it in cell culture, and then we move to mice. If things don't work in cell culture, we don't move them to mice. If they don't work in mice, we don't also move them to large animal models. So this is just to show you some of the results that then led to transferring or starting experiments in large animal models, specifically the Tay-Sachs sheep. So we started a model where mice, GM2 mice, are treated neonatally. And you can see a little picture of a neonatal mice over here, very cute on the left bottom corner. And what we found after a neonatal treatment, so immediately after birth, about day 0 to day 1, they received 2 injections into the CSF, into the lateral ventricles. And what we observed was a dramatic increase in survival that you can observe here or appreciate here on the left side, the panel. The red line is the survival of the AAV treated animals. And then as a control, you have wild type or heterozygote animals. So essentially they essentially survive to normal lifespan of animals, not all of a normal animal, not all of them, but a good percentage of them survives for a very long time with very good function. So one of the tests that we do to test motor function and coordination is this rotorod. It's a rotating dowel. You can see a little cartoon over here of a poor mouse kind of hanging for dear life. And so we test them. This is a simple test. Actually, it's not painful for them. And they just run along until they can't any longer run and they fall off. So what you can appreciate in this graph is that animals that are untreated, so GM2 animals, says GM2 at 120 days, they essentially can't perform. The moment the dowel starts rotating, they essentially fall off almost immediately. As contrast, you can see here the animals that were treated with the AAV on the left side of the graph. You can see here that 120 days, 150 days, they really maintain normal performance compared to wild type animals. Heather, if you could move on to the next slide. Not surprising, when we measure GM2 levels in the cerebrum, the cerebellum and brainstem, we've essentially normalized GM2 content, GM2 ganglioside content in the brain, which is the biochemical, the lipid that accumulates in the brain of Tay-Sachs and Sandhoff patients and in the animals as well. So you can see here that the levels in treated animals, AAV9 treated animals, essentially comparable to that found in normal animals. And of course, much, much, much lower than that is observed in GM2 mice that are untreated. Next slide, please. So after succeeding in treating neonatal animals, we then decided to move the therapy in a little bit more stringent manner, where we actually inject 4 to 6 week old animals receiving another delivery modality into the cerebrospinal fluid through the intrathecal space. And what you can observe again here is that, in fact, the survival is also considerably expanded over that of untreated animals. So the red line is that for treated animals, and untreated animals is black line, and they died about 120, 130 to 140 days of age, while the treated animals survived considerably longer. Similarly, again, the same test, the rotor-rod test, which is the motor function and coordination, is the animals were tested at different times after treatment, and at different ages actually, or different times after treatment. And you can see here that they maintain the performance at the 120 days, where animals can no longer perform the untreated. And, you know, over time, that performance seems to decline a little bit. And that has to do with the challenge of delivering into the CSF of mice through the intrathecal space, which is quite challenging. So with this data then, Heather, that is the expert in large animal model experiments, decided to take this on and move it towards the sheep, and that's what she's going to be presenting now.
Hi, okay. So Tay-Sachs disease, ooh, there's an echo. Tay-Sachs disease in sheep is a naturally occurring disease. It's not unlike late onset Tay-Sachs in that not all sheep get the same clinical signs, and they have a protracted lifespan. They can live to sexual maturity, and they can survive about a long time. We tend to put them down about 9 months of age when they lose the ability to stand. But the most common things that happen with late onset Tay-Sachs sheep is the problems with their feet. They don't really know where their feet are in space, and so they have them knuckled, or they go in opposite directions, and then sometimes they have seizures as depicted below. And so what we're doing in these sheep is we're using them as an opportunity to test therapies for late onset Tay-Sachs and Sandhoff patients. And so the way we do this is, because sheep are so large, they're more similar in size than humans, than any other laboratory model really, we're using these animals to develop this therapeutic. So Tay-Sachs is kind of somewhere in between juvenile and late onset, but they have a significant amount of disease associated with their forebrain or their cerebral cortex, which is not necessarily present in late onset Tay-Sachs or late onset Sandhoff disease, or if it is, it's definitely to a lesser degree. And as you know, most of the disease in late onset Tay-Sachs and Sandhoff disease is in the cerebellum and the spinal cord. So we did a study in the Tay-Sachs sheep with the desire to develop a therapeutic for late onset Tay-Sachs and Sandhoff disease. But in the process, we recognized the fact that what may work the best in sheep may actually not necessarily, it may be too invasive or more invasive than what's necessary for late onset Sandhoff or late onset Tay-Sachs disease. So I just want to start with that before I start talking about the therapies and the effects in the sheep, that we may have to be more aggressive in treating sheep Tay-Sachs disease and treat than we would have to be treating the human forms. Okay. So what we did in this study was we tested injecting into the cerebral spinal fluid, which is the fluid that surrounds and goes inside of the brain. And its major job is to kind of help clean out things that need toxic substances that need to be cleaned out, and also protects against concussive forces. And what we're doing, like, you know, if you get in a car accident or something, and what we're doing here is trying to use this cerebrospinal fluid, which is just kind of water surrounding the brain and the spinal cord, as a delivery mechanism for the therapeutic. And so what we did in these studies, we injected sheep in 2 lateral ventricles. They're in the middle of the brain on both sides, right and left. And they're just little fluid pockets, and that's where CSF is, one of the areas where CSF is produced. And then we're also injecting in the cisterna magna, which is kind of at the base of the neck, and then the lumbar intrathecal space was just towards the back. And this is because when we really wanted to test to see if CSF delivery was going to work, we gave it the best chance we could. And so we tried every possible delivery route for CSF delivery. And then we also tested just cisterna magna only delivery, which is just at the base of the back under here underneath the cerebellum, because we wanted to really understand what the distribution would look like in the Tay-Sachs brain, because this particular delivery route can be really, really efficacious for treating the cerebellum and the spinal cord. And so this is just kind of the summary of the data here. So the untreated Tay-Sachs sheep are in green, and every drop of the line represents a death of an animal. And you can see that we euthanize these animals in a pretty narrow window. When we treated by the CSF delivery that has this lateral ventricular cisterna magna and the lumbar intrathecal, which is the purple, you can see we've lost 1 animal so far, and that animal actually died from pneumonia, unfortunately. So it wasn't necessarily died from neurologic condition. And then the remaining animals are depicted by these kind of hexagon shapes up at the top. at their different ages. So they're all at least 2 years old, and 2 of them are over 3 years of age. And then we have 1 animal ongoing still for the cisterna magna only group. And that was the one that was just treated towards the back of the brain alone, which we believe is a good location for treatment for Tay-Sachs or late onset Tay-Sachs and late onset Sandhoff disease. So these are all of the sheep here. There are little headshots across the way. We have several sheep that are a couple greater than 3 years old, and then we have some that are greater than 2 years of age. And the one cisterna magna only animal here is still ongoing at greater than 2 years of age. And that's number 261. He's kind of everybody's favorite. He's very, very friendly. So I could try to describe this, but I think videos speak 1,000 words better than me describing it, but the sheep are actually doing really quite well. I hope this video isn't too choppy for you, but this is one of the CSF treated sheep, 212, who is now greater than 3 years of age. Normally they reach humanum point at about 9 months of age, and you can see that 212 isn't perfect. She kind of has a little bit of a hitch in her step, but she's definitely one of 2 of the longest lived sheep that we have had. She has some difficulty getting around with gait, but otherwise is in great body condition and is doing really quite well. And then this is 212 on the right-hand side. And he is a cisterna magna only treated sheep at 2 years of age. And you can see he has also a little bit of a wobbly gait, but is getting around and moving quite well. And he is maybe the happiest sheep I've ever met in my whole life. He's very friendly, and he really enjoys life and his friends around him. So this is what the Tay-Sachs sheep look like now. They're still doing really well. And so we're really enthusiastic about moving this therapy forward for treating late onset Tay-Sachs and late onset Sandhoff disease. So we wanted to try to understand what it looks like as far as therapeutic. And so this is pictures of the MRI. You can see that the normal sheep brain MRI, the inside of the part of the brain is black, the outside is a light gray. You can see that that's kind of blurred with the untreated Tay-Sachs animals. And then you can see with the CSF animals, it's largely normalized, which is important. At least the brain looks quite normal. And then also we collected CSF from these sheep at different time points. GM2 obviously is a storage product. We can measure that in the cerebrospinal fluid quite well, and you can appreciate, I hope, that the red boxes are a lot higher from the untreated animals than the green in the normal animals. And then the AAV treated animals in different ages are depicted down below in the purple dots, suggesting that the GM2 storage in the cerebrospinal fluid of these sheep is normalized, which is really an important biomarker for clinical trials so that they have something to try to sample. I mean, it's easy to do an LP in a patient and be able to say, okay, this is CSF is ganglioside is being cleared or not cleared. And so with the increased survival in the Tay-Sachs sheep over now longer than they've ever lived before, and the efficacy in the mouse studies, and the biomarkers that look really quite promising, we've moved forward with trying to move this towards clinical trial. And so UMass Medical School, Miguel and myself have, and Cindy Tift and some other collaborators have put together a document that's been submitted to the FDA, trying to move forward with a gene therapy clinical trial for late onset Tay-Sachs and Sandhoff disease. And so the first step of this is to schedule a meeting with the FDA, and this is done by sending in a pre-meeting request form. And so we've submitted the request, and that has been done. So we expect to hear back from them in a couple of weeks, and then we will schedule a meeting with the FDA to try to figure out clinical trial for late onset Tay-Sachs and Sandhoff disease. So that's where we are right now. We anticipate to hear back from them in the next sometime this summer, and then we will know more about what we need to do to get this to late onset and Tay-Sachs disease. Miguel, you want to add anything to that?
No, I mean, it's exciting to be at this stage, and certainly we hope to move through it as fast as we can to initiate a clinical trial in late onset. All the data suggests that it can have a profound impact, so we're quite excited to be at this stage and move it forward. So yeah, that's about it.
Great. Well, thank you, Heather and Miguel. I'll open up for any questions people may have. And while people are writing them down, maybe I'll just start off with a couple of questions. So could you talk about how you translate your dose and your route of administration from the sheep to humans? You showed a couple of different potential routes of delivery, and do you have one that you think is going to win out over the other?
Yeah, so cerebrospinal fluid delivery is definitely the one that we're pursuing. There's a couple of reasons why. It's directly in the area where we need to treat, which is the central nervous system. It doesn't require parenchymal injections like is being done in the infantile and juvenile patients where they're getting injected directly into the brain. And we think that even less invasively, we would be able to just inject the CSF behind the brain using a catheter delivery method that I presented last year instead of having to go into the brain and directly inject into the lateral ventricles, which is safe in human patients. They do it all the time, but it is more invasive than perhaps is necessary for late onset.
Great.
I wouldn't want my brain injected if I didn't have to have it. So I think that everybody else feels probably the same way.
And how does the size of a sheep brain compare to a human? Are you thinking that you would use similar doses?
Yeah, so we calculated the dose based on brain weight, and we scaled up from the brain weight of a sheep, which an adult sheep, the time of treatment, these sheep have about 80-gram brains, and the adult sheep is about 120-gram grains, and then an adult human is around 1,400 grams or 1.4 kilograms. So we just extrapolated the dose from the sheep up to human patients based on brain weight, which a lot of other, that's pretty common in the field to do it that way.
Okay.
And people come in all different shapes and sizes, but the brain and the spinal cord is pretty consistent. So we feel like that's better than dosing based on body weight.
Okay, great. I have a couple of questions that have come in. First question was, who would be the patients that you would first test this in?
Late onset Tay-Sachs and Sandhoff disease patients would be the patients that would be included in this IND. So we would not, in this first, there is an IND, there's already a clinical trial going on that's from our group for infantile and juvenile Tay-Sachs disease. That is, as many of you know, is already ongoing. This one is specifically for late onset Tay-Sachs and late onset Sandhoff.
Okay, and for someone who undergoes the treatment, what kind of recovery time would you anticipate after the administration?
So the catheter-based delivery method is done very quickly, and I don't believe there would be any recovery time required. Miguel, do you have a comment to add to that?
No, I think that's done pretty much as an outpatient treatment. I think the question is not necessarily related to the procedure itself, but probably response to the therapy. And the response to the therapy is very difficult to quantify, I would say. I mean, Heather has data showing that biochemically can occur very rapidly, right? But in a human, I mean, that's really difficult to quantify how long a human would take to start responding to the therapy. If it's anything like some of the experiments Heather has done, it could be in a couple of months. But we've also seen in other trials that it takes 6 months to start to see some effects. So it's really hard to predict based on animal experiments what's going to happen in the human. Well, first of all, I guess you have to start with the basic question: does it work, right? And once you find that answer, you also find how long it takes for the patients to respond. But based on what we've seen in several trials, including some of that were somewhat involved in, patients start to show some responses within maybe 2 months or so. And then, some of them a little bit longer than that, to 6 months and beyond that. So if it follows the same pattern, I guess that's sort of what could be expected in terms of starting to see some degree of response.
Right. A couple of other questions. One is, why are the infantile patients receiving intraparenchymal injections, but not the older patients?
The disease in the infantile patients actually starts, and Camilo, feel free to join in because I know this is your area, but the diseases are very different in the infantile and juvenile patients as late onset patients. And so that study was designed to treat them, and it did require injections into the thalamus. We don't believe that's required for late onset Tay-Sachs, and it's more invasive than we need. And Camilo, please feel free to comment.
So the feeling, and I think Heather is 100% correct. The disease has different manifestations in infancy and in adults. In adults, the majority of the at least symptom origin seems to be localized to primarily to two brain areas. One will be the spinal cord, the other one will be the cerebellum. And they believe that if one were able to help those areas, that you may already experience some degree of benefit without having to be over-invasive.
Great. And then maybe just one last question. There was a question about the blood-brain barrier, and is this a challenge for this type of delivery?
No, so you could think of it as we're doing it on the other side of the blood-brain barrier, right? By going into the CSF. So we're on the other side of it, if you will. So it is a challenge when you want to do it, injection into the bloodstream. Then the blood-brain barrier is definitely a barrier. Just one thing that I just wanted to clarify, maybe, well, Sue reminded me of this in the comments, and rightly so. We're nowhere near ready to start recruiting patients. This is sort of submission to the FDA is very much the first step. And then there's still a lot of studies that have to come through that are essentially mandated by the FDA to ensure that there is a high degree of safety to whatever it is that we're going to do in human, right? So just to clarify that, recruitment is not about to start tomorrow. We wish it did, to be perfectly honest with you, but it's not. So this is still going to take some time to be in that stage where the trial is approved by the FDA and it's really then ready to start. And again, you'll never see me or Heather involved in any of that, because then it's totally on the clinical side that make all the decisions in terms of enrollment and so on. So we'll disappear out of the picture completely, and we hope that to disappear very soon.
And to that end, there's not going to be any reason why you cannot participate in the ongoing clinical trials. It's not going to be an exclusion criteria. You can continue to participate in the things that you're doing now. And please don't stop. Keep doing what you're doing, because it's, probably it's going to take a village to treat late onset Tay-Sachs, and it might take multiple therapies to make it actually really efficacious. So please continue to do what you're doing, and don't feel like we're going to exclude you because of anything that you're already doing. Okay.
Right. Maybe I'll just combine the last two questions before we move on. One is, would you anticipate this being a one-time treatment? And for people who do have issues with balance or muscle strength, would the expectation be that you might see some improvement in those areas?
We hope so.
This is always the $1 million questions, and we sure hope so.
And actually, the clinical evaluation parameters actually look at those as potentially as outcome measures. So we certainly will be looking at those kind of things to demonstrate efficacy.
So one of the things that we don't know right in humans is, especially in late onset Tay-Sachs patients, is that the symptoms that one has, are they caused by losing cells or dysfunction of cells that are still there, right? We know that it's a party is losing some cells, but it's also possible that quite a bit of it is due to cells not working properly, right? So if it's the last case is more prevalent than what we think, then I think there is a good chance of actually reversing some of these phenotypes, right? But that's really, unfortunately, we can't reassure anyone that that's going to happen. That's what we hope it will happen. That at a minimum stabilizes where people are now and don't get worse. But we may get surprised that in fact people actually improve. So I would say that's the best answer we could give now to this question. And it's one that we're asked all the time for obvious reasons.
And with this being a gene therapy, you would anticipate a one-time treatment?
Certainly for the CNS, for the brain, yes. Other organs, the jury is still out on that one. But in terms of the brain, right now, our expectation is the one-time treatment, yes.
For late onset Tay-Sachs in Sanofi, we're expecting one time only.
Great. Okay, well, thank you both very much. It's really exciting to hear that there's even more advances now going on that may find their way to the clinic. Our next speaker is Camilo Toro, who's going to talk about current